excited to have you here on MyFlora Fridays, which is a new webcast that I decided to do in collaboration with MyFlora DNA. And I figured I speak to a lot of cannabis people, experts, scientists, all kinds of folks every day. Why not record our conversations? So I'm so excited to have you here. I was just going through your literature on Google Scholar, and there's definitely things that I would love to have you talk about. But first and foremost, an introduction. So I'm going to go ahead and let you speak for yourself, introduce yourself, tell us what you do and who you are.
Sure. First, thanks for having me. Great to be here. A little bit of background about myself, I guess, is I from Ontario, Canada. Pretty typical childhood. Grew up, went into plant biology, so I did my bachelor's at the University of Guelph in plant biology. My master's was in tissue culture, working with echinacea, and so I've been working with medicinal plants for a long time, for about maybe 20 years, give or take. And the goal of that was really this is going to sound really very similar, is that our goal is really to develop methods for the standardization of echinacea. And so all medicinal plants have similar challenges, and that's kind of where I got my start. After that, I started a PhD in Mississippi, but I dropped out of that. So I am a university dropout. But then I got back on the horse and ended up doing a PhD at the University of British Columbia. That was working with breadfruit, a very different direction. But after that, I kind of returned. I came back to Guelph as a postdoc, ended up as a faculty, and started working with cannabis around 2015. And just to give a little bit of background about that is that in Canada, the medical cannabis was legal since I think about 2001, but it wasn't really a commercial thing. So there was only one producer in Canada, and then you could grow your own as a patient. What happened in 2014 is not the legalization, but the commercialization of cannabis. And this is when commercial production of medical cannabis started. And so I was a postdoc at the time. I actually submitted a license application. So I was going to start an LP, but then I got a faculty position. After that happened, so the timing was just so that there was a lot of money flooding in and investment. So there was a lot of potential support for research, and we got our start in working with companies. I got my first license in 2018. And so this was just before legalization of recreational cannabis in Canada, sorry. And I've been kind of going ever since.
Okay. Interesting. I also was a college that was during my bachelor's degree. I went to Colorado State University and did 5 full years, full semesters, full loads, summers included, because I was taking all kinds of classes, drugs in the human body, sex and sexuality, all kinds of stuff that was just really interesting to me. Couldn't pass organismal biology. I took that
Mm.
3 times before I finally passed it, but that was after I took a hiatus and went back to school to get my bachelor's and then my master's, and then finally started my PhD in cannabis after Colorado legalized adult use in 2012. And then in 2015, I decided I wanted to do the PhD. I had to find somebody who would let me do a PhD in cannabis because, unlike Canada, the United States still really doesn't allow research on cannabis at academic institutions. There's some of them are doing research on hemp, but I'm really one of the only people that's actually been able to research THC type cannabis that's available on the legal market. So I think we started in cannabis around the same time. So we've kind of been going at it together. And I've been looking through you can go on Google Scholar. For people who don't know, you can go on Google Scholar and you can look up not only articles, but you can also look up an author. So I'm looking at your incredible list of publications, and there's so many things on here that I would love to talk to you about. But you know your research better than I do, and I can see here that you are involved with tissue culture, epigenetics, polyploidy, somatic mutation accumulation. So I'm not sure where to start, but, like, what is your focus right now? Like, what's your passion right now? Or maybe where did you start and how did yeah, let's do that. Where did you start? What was your passion to start with that has brought you kind of and your journey along to where your interests lie now?
Yeah, so my background is really primarily on plant tissue culture. So that's where we started. And I'd say there's a couple of reasons for that. One is that that's my core focus. But the other one is that from a licensing perspective, tissue culture was much easier for us to get. So we were able to get a license to do tissue culture because we weren't flowering out mature plants, and the security requirements were much more reasonable. And so that was a better starting point for us. And so, like I said, in 2018 is when we got our first license. Before that, I was working with some companies also doing tissue culture. Our initial start was the goal was to develop somatic embryogenesis technologies for a number of reasons. One of my main interests has always been synthetic seeds. And so theoretically, you can produce clonal seeds using tissue culture technology. Unfortunately, cannabis is not the most cooperative plant in tissue culture. So that's kind of still a long-term goal, but no one's really crossed that barrier. So, we've done a little bit of somatic embryogenesis work and we've had some regeneration, but it's really not a reliable system. So that's kind of up in the air. That being said, we worked on that for a while. So we were developing micropropagation. I'd say in the early days, there weren't that many publications, and the publications that were available, we didn't find were very reproducible. And I think a lot of people found the same thing. So we tried to use the methods that were in the literature, and it failed horribly. So we spent quite a while just working on the basics of tissue culture. Once we had that up and running, then we sort of diversified and started getting into more of I wouldn't say breeding, maybe pre-breeding is the better term. So we do a lot of polyploid work, and we are doing some breeding work, but it's more foundational than that, I suppose, largely. Other than that, we're doing some other interesting things within tissue culture. Most of the epigenetic and somatic mutation type work I do through collaboration. I've worked very hard not to be a molecular biologist, so I do that largely through collaboration. And I can speak to it, but that is not my expertise per se. So that's kind of where we are. As far as the most recent projects we're working on meristem culture methods for virus eradication, or viroid eradication in this case. And I know there's a lot of people doing that already, but I think there's a lot of room for improvement. So we're going down that tunnel.
Okay. And so, all right. So that's really the using tissue culture to remove pathogens is something that is like it's a big conversation right now, because hop latent viroid is pretty prolific in many facilities, and the idea is that you can clean your mothers and maybe keep a supply of stock in the form of tissue cultures, kind of like a bank, like it could, because
Yeah.
tissue culture takes up very little room in a fridge. You can have many plantlets or whatever you call them, and switch out your stock so that you have a fresh supply constantly through a tissue culture bank, I would guess, and then have multiple different strains. Where if you have like a mother room, you need to have all of those mothers, keep them in veg. They take up lots of space. They take up lots of resources in the form of water, nutrients, manpower, lighting, all of that stuff. So, can you talk about the pros and cons of keeping, a stock of tissue culture and however many strains, and then how successful is their permanent, or because I don't, yeah, how successful is the removal of pathogens from with using tissue culture, or do the pathogens have a tendency to, like, you're decreasing the viral load and they can come back after you generate them so many times?
Yeah, those are good questions. I'll start with the first part. And I think one of the things that's really important for cannabis and tissue culture and more broadly is that, not every plant needs to be tissue cultured or micropropagated. When we first started, a lot of people were looking at like large scale propagation based on tissue culture, but in reality, cannabis is so easy to take cuttings that large scale micropropagation may not actually make that much sense. But even in cases like that, tissue culture has a role. And I think it's always good to look to other crops. So in the case of clean plant programs, we can look to other things like, I think the best example that I can think of is potato. And, the cheapest way to plant a potato is to, chop up a tuber and plant it in your garden. But the problem is that those tubers will be covered in all sorts of pathogens and you're going to have sick plants, right? And so what the potato industry has done is they've established a clean plant program where all potatoes start with tissue culture. They go through the virus eradication, testing, certification, all of that. And what they do is they plant out these tissue culture plants that are known to be clean. So they're certified virus and disease free. They multiply those in a greenhouse, then they eventually multiply them in the field, and then eventually the potatoes get planted by a farmer in the commercial field, right? And so by doing this, what they've done is they get most of the benefits of tissue culture, but they don't have to bear the full cost of tissue culture. And so I think it would, the best approach for cannabis is to probably come up with a similar solution where, yeah, you have your tissue culture, clean plants in tissue culture, and then every once in a while you replace your mother, you propagate it for a certain period of time and produce flower, and then you replace your mothers on a routine basis. So you don't have the accumulation of disease and stuff. So I really do think that's the sweet spot. And so you get most of the benefits of tissue culture, but you don't have the full cost of tissue culture.
So you're balancing between having a mother room and also having a clean stock to replenish every
Yeah, some
however many.
and this is all obviously this is a business decision. Some people will say, well, the value of clean plants coming out of tissue culture, I'll just propagate through tissue culture. If you're willing to spend the money to do that, then maybe that makes sense. But from my experience, just because cannabis grows so quickly in the greenhouse or growth room, and you can propagate it so easily that tissue culture really, it's hard to justify the cost of tissue culture. But for maintaining mothers, so your gene bank, tissue culture makes a lot of sense. And we've done some cryopreservation work, so you can put it in liquid nitrogen and store it long term. But even just in tissue culture, if you put it in low temperatures, so we have two culture rooms. One is at 27 degrees Celsius. The other one is at about 11 degrees Celsius. So it's like, I think, 50 Fahrenheit. And at 50 Fahrenheit, the plants basically grow really, really slowly. So we can keep them in there for, up to 8 months without touching them, like no watering, no fertilizer, no pest control, no labor, nothing. Then we can bring them out and they bounce back out. We subculture them, put them back into the fridge. And so for preserving large numbers of genetics, tissue culture is definitely the way to go, in my opinion. And I think their clean plant programs is definitely the way of the future. I think we're to the point with hop latent viroid that it's absolutely everywhere. There's evidence that it infects all sorts of plants, not just cannabis, not just hops. And so you can't avoid it, right? You can't eradicate hop latent viroid. So all we can do is manage it. And the best way to manage it is to start with clean plants, and so that it never accumulates to a level that causes economic loss, if that makes sense. As far as the eradication goes, this is actually probably the more interesting part of the question, in my opinion. And that's that, without getting too deep into the weeds, the theory behind meristem culture that I was taught in school and most people are taught in school is that essentially that the meristematic region is not connected to the vasculature of the plant. So the virus has not had a chance to make it to the meristem. That's part of the story. But if you dig deeper, there's a bunch of other things going on. And there's a couple papers from around 2020, I believe, from a Chinese group that found that there's a gene expressed only in the meristem. It's the Wushel gene, and it has direct antiviral activity, right? And so the meristem may not be connected to the vasculature, and that probably is part of the reason meristem culture works. But there's also antiviral components in the meristem itself. And what I find really interesting about this is that from what I was reading, the mode of action is that it prevents the virus from forming a protein coat. And where that's really important in this conversation is that viroids don't have a protein coat. Right. And so my hypothesis in this is basically that meristem culture will be less effective for viroids than it is for viruses. And if you look through the literature, that kind of comes out, meristem culture is fairly effective for viruses, but it's a less consistently effective for viroids, right? And so, I can't get into too many details, but we're looking at alternative treatments that we can combine with meristem culture that will make it more effective for viroids. Back to your question about whether it actually gets rid of all the viruses or viroids, or if they're just like at really low levels and they come back later, I don't know.
Depends.
Yeah, I mean,
right.
I know a lot of people are offering meristem services, but I've heard many, many stories where people get so-called clean plants out and then they become infected later. Did they get reinfected, or did they always have the infection? That's the big question. And I don't know.
Yeah, because I think of, people, right? If you get chicken pox when you're a little kid, the chances of it hanging out dormant in your nerve cells is pretty good. And you might develop shingles later in life. It's the same virus that's just been chilling until it becomes active again. People with cold sores, right? It goes away and then you get stressed out and it comes back. And yeah, viruses are tough. And I imagine it's sort of the same for plants. And I think it's interesting, the idea of cleaning plants. I have to wonder, like, is it really cleaning it, or is it just lightening the viral load to such a point where it looks clean because the tests come back clean because they're below the limit of detection, or
Yeah, and
they don't amplify well in PCR.
I would say like these tests are pretty sensitive, but
Mm,
the other tricky part about this is that when you test a plant, you're not testing the whole plant, right? You're taking a
yeah.
sample of leaves or whatever, and you're testing the leaves, and then you're trying to declare that the whole plant is free from the virus. And in the case of hop latent viroid, we know that it
Mhm.
accumulates in some tissues more than others at different times in the life cycle and whatnot. So you might test some leaves, maybe they come back clean. Those leaves might have been viroid free. That doesn't mean the stem is viroid free, right? And so
Or the
there are
root.
problems. And the other fact, and this is what you were getting to, is that it's latent viroid, right? And the word latent is very important here because it can survive very low titers for a long time and then flare up, right?
Mhm.
So it's kind of like the cold sore situation. So it's very complicated. And I think, there's a couple articles on meristem culture, but I just think there's a lot more unknowns than there are knowns in this situation. And, I don't think we're there yet. So I'm pretty excited to try to take that on. I also think, there's a lot of different companies offering these services. I've seen pictures on the internet of what people are calling meristems. Sometimes they're meristems, sometimes they're shoot tips. And if it's a shoot tip, it's probably not going to work, right? And so it's really important to know exactly what they're doing and how they're doing it to know whether or not it would likely have the outcome you're looking for.
So there's different kinds of tissue culture, right? Is what you're getting at there. Like you're talking about meristem tissue culture, which is so the meristem is located where on the plant?
So any given plant has many meristems, but
Mhm.
basically, all new growth in plants comes from a meristem.
Okay.
And so every shoot tip has a tiny little meristem inside, right? And so what I was getting at is that like sometimes when people take a meristem to get a clean plant from a meristem, you really need to make sure you have only the meristem. If you take too much tissue, there's probably going to be viruses, right? And then you're not going to get a clean plant. You might get a plant with really low titer rates that looks clean, but you're not going to get many clean plants out of that in the other end.
Okay.
So there are many different types of tissue culture. In this case, I'm just talking about the explant size. And so
Mhm.
what size of tissue are you putting into culture? And that's going to have a huge impact. And I know when you go right down to a true meristem, it takes a long time to come out because it's a very small tissue and it just takes its time, right? And so I know there's an incentive for companies to take larger pieces of tissue because they get plants
Mhm.
out quicker, but quality versus quantity, I guess.
Okay. And then I was watching Chad Tierney doing some tissue culture in his lab at Colorado State University, and he was talking about he does kale, and he's got like a petri dish with multiple little cultures in there, and he talks about callus. And so what is the difference between the meristem tissue culture and what he's doing with the rooting out or developing a callus and then rooting that and then having that as, I guess, the explant? I don't really know the process.
Yeah, so I mean, so callus is it's the definition would be like a mass of undifferentiated cells. So one of the interesting things about plants is that you can have a leaf cell that can change its identity. So you have a leaf cell and you can deprogram it so it's no longer a leaf cell and have it multiply as callus, and then it can regenerate into a meristem or a root meristem, shoot meristem, or somatic embryo.
And is that done with hormones then? Or
Yeah, yeah.
yeah.
In general, so if you're producing callus, basically what happens is that you're taking whatever cell you started with, you're de-differentiating it, so it loses its identity,
Mhm.
and then it multiplies as just a mass of cells, right?
Mhm.
If you get deeper into the conversation, callus is not truly de-differentiated. Callus, there's many different types of callus. Some of them have different regeneration capacity. So when we say de-differentiated, that's a bit of we're glossing over the reality, but
Mhm.
that's basically what it's defined as.
So
And
maybe
so,
like erasing a chalkboard, like most of the words are gone and stuff, but there's still some chalk on the board.
yeah, or maybe you,
Or
you missed a few words.
Or you can kinda still read it because you can even though the chalk's gone, if you get in the right light, you can kinda still see it.
Yeah, and they have all sorts of words for the different types of callus. So sometimes they'll call it like embryonic callus, and um, there's a bunch of different words. Or sometimes they'll refer to cells as competent, which means that they have
Mhm,
the capacity to regenerate. I think that's a bit of a cop out because I believe all cells are technically competent. We just don't know how to unlock that potential, right? And in the case of cannabis, It produces a lot of callus. You can produce callus really easily, but regenerating from callus is extremely difficult. And so some plants, you can produce callus and you can produce millions and millions of plants because you have millions of cells, right?
Mm.
That's kind of the theory and the potential of callus. But in the case of cannabis, the reality is that we can produce lots of callus, but to get it to turn into plants is extremely difficult.
Okay. So your success rate of, so I was talking to Bill Graham, who I think you talked with yesterday as well. He made me a little, I'll show you. It's a
Yeah,
little
yeah.
plantlet that he made at a table at a restaurant of all places. But so you can produce like you can put millions of things, tissues into tissue culture and hold it in suspended animation in this gel for however long. Every now and then, I guess you have to replenish the gel because it can't, live there forever with nothing. But where the challenge really lies is pulling it back out of that suspended animation, getting it to root, and then having it strong enough to transplant and then grow into a plant. That's coming out of it is the challenge, right? Is from what I understand.
Yeah, so that phase is usually like called acclimatization, where you're transferring from tissue culture into the real world, right?
Mhm.
My my personal feeling on that is that if you have healthy plants, then the acclimatization is actually quite easy. A lot of people when they're having problems with acclimatization, they start playing with the humidity and the light intensity and the temperature and all of those things, right? But I think in reality, the better approach is to take a few steps back at your tissue culture methods to make sure that the tissue you're producing that's going into the greenhouse is robust and healthy, if that makes sense. And so, a lot of the time, and it's the same with cuttings. Like if you're taking cuttings and they're not rooting very well, a lot of people will be like, oh, well, optimize the temperature of your substrate, or, it's the humidity in your domes, it's blah, 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 right? I would argue that it's more effective to go back and say, how can we make our mother more healthy? Because if you have a healthy mother plant, it roots extremely easy. If your mother plant is sick, you're going to have problems rooting.
Okay. So
And
so go
so I
ahead.
just think people tend to focus on the wrong part. They focus on the environment at that time instead of looking at what did you do that led up to that.
Okay. So in light of that, it's really if you're thinking about banking your stock for the future to ensure that you've got things for the future. It's better to do it at the get-go before you get hop latent for or whatever, before you get any
Definitely.
problems instead of the other way around, like, oh no, we've got these problems. Let's go ahead and get some stuff into tissue culture so then we can get rid of this room and start over because those clones might not actually come out on the other side being okay because they weren't very okay to begin with. Is
Yeah,
that
that's
okay?
definitely true. And to that point, we for a lot of our pheno hunting, we germinate the seeds in tissue culture and then we multiply them there and then send one to the growth room to be screened. And so the backup we have is in tissue culture already. It's never been out of tissue culture. And then we screen a copy of it. And then once we find the winning copy, we already have it in tissue culture. And
Okay. That's an
it's kind of
interesting.
flipping it on its head. And so the nice thing about that is like if you do a pheno hunt normally, you have to grow the plant until it's large enough to take a cutting. You have to take that cutting and maintain it as a vegetative plant in another room, et cetera, right? If you do it the
Mhm,
way I'm talking, you germinate the seed in a test tube. Once it's big
mhm.
enough, you take the top and transfer it to a new container. You take the bottom and put it to the growth room. And then you don't even have to have a secondary room to maintain the vegetative plants, right? You just have it essentially in the low temperature culture room. And when you need it, you just bring it back out, right? And it's disease free, virus free, all that stuff.
So then you would have okay, so you have a seed, you germinate it, you split it up. One goes to the pheno hunt and one goes into storage
Stays in tissue culture.
so that when you figure out which one or ones you want to keep, you can go back to the fridge and be like this one, that one.
Yeah, yeah, exactly.
Okay.
And
Gotcha.
so,
Okay.
the floor space it takes is much like dramatically less. The labor it takes is less. And you can take it one step further, actually, is that you don't even need to let the seed mature. So you can do embryo rescue where before the seed is fully mature, you can dissect out the embryo and put that in tissue culture. So before the seeds are even mature on the plant, you already have it growing in tissue culture.
Interesting.
Yeah. And so, the long-term goal of this is for speed breeding. The reality is that when you do embryo rescue, I think with some work, you could save time, but a lot of the time with embryo rescue, when you put it in culture, it takes a little while to establish, but it's still a viable approach.
So is that like taking the jacket off the baby?
I guess so. It's kind of like doing a cesarean section and taking a preemie out and incubating it.
I always think of seeds as like little babies that are I in a jacket, but, like, with their lunch. They've got their box lunch in there, so it's like taking the
Right.
baby out
Yep.
and leaving the lunch and the jacket behind.
Well,
Is
you're bringing the lunch with it, right?
Oh, you bring the lunch with it? Okay.
Yeah. Yeah.
And I imagine that takes seeds are tiny, so you work under some sort of magnifying glass. I imagine it takes some sort of skill to be doing all these tiny little things with
I with seeds, I mean, they're large enough that you don't really need. You can do it under a microscope, like a low mag microscope, or you can do it on the bench, like in a
with
flow bench.
with reading glasses on or something.
Yeah.
My eyesight's terrible. I don't know if I could do it without, like, a dissecting scope or something.
Well, like
Uh,
one of the advantages of doing this is that it's actually quite easy to sterilize seeds and bring them into culture. It's actually a lot less work to do that than it is to take shoots and bring them into culture. And so by doing that, we can get all of the plants in tissue culture without much work. And then we have them in tissue culture. So we save a lot of time and effort with the maintenance of the backup plant. And then we do the pheno hunt. We don't always do it that way, but I like to do it that way.
that's your favorite way?
Yeah.
Oh, okay. Now, I have had a theory, and I don't know if it has any water, but you would be the person to ask about this. So, I've been really interested in epigenetics for quite a long time, and especially in clonal organisms, plants that and cannabis is a perfect model or organism to look at this because that's the way we generally propagate it. But I have this feeling that the stress of going through the cloning process for both the mother and the resulting clones is somewhat stressful. Like, plants respond to their environment, they respond to insects chewing on them and things like that, so or being broken or whatever. So I have a feeling that some of the changes that growers see when they have a mother and they do multiple clone runs or are cloning from clones, they start to see some sort of change in their plants or some sort of deleterious sort of, I don't know what the word is off the it's Thursday. I just got back from a conference, but you know what I mean? Like, the plant, it's just not the same as it was, or they're not growing as vigorously as they used to. They're not producing as much as they used to. Their terpene profiles have changed, or whatever the case may be. And, I always thought, like, well, really, the genome hasn't changed because they're clones. Some of your research talks about a somatic mutation accumulation, but also, like, the epigenetic changes in response to stress, which is how plants respond, can also lead to these changes. So, can you talk about both of those things?
Yeah, and I'd say there's a third one as well, and that would be the accumulation of pathogens and things of that nature. So there's that's the third one. But yeah, I guess I'll start with the somatic mutations. And we did a study where we tested different parts of a single plant and compared them using like really deep sequencing. So we were looking for small mutations, right? And basically what we
Mhm,
found is there are indeed mutations that shouldn't be a surprise to anyone.
mhm.
You could argue about how many mutations you would expect, but the fact that there are mutations is pretty standard understanding. So whenever cells are dividing, DNA replication is never perfect, right? So you're going to have some mutations. And what we see in plants is that this is pretty well established. And if you read that paper, we cite what's known as Mueller's ratchet, and this is basically a hypothesis that over time organisms will decline because of somatic mutations. And basically, the theory is we know mutations accumulate. plants continue to divide throughout their life. So they're not like animals where we divide a certain number of times and then we're pretty much, I'm full grown.
As big
I'm
as
not,
you can
my
get.
cells aren't really dividing actively. Plants are very different. They continue to divide forever, right? That's how they grow. That's how they live. And so they're going to have a continuation and continued accumulation of mutations. And basically, what Mueller's ratchet suggests is that most mutations are negative, right? It's a lot easier to remove a gene's function than to enhance a gene's function. So most mutations are bad. And so over time, we're going to have an accumulation of bad mutations and we're going to lose vigor. And, this isn't just cannabis, right? So, it's not some crazy cannabis thing. This is really well known from a lot of plants. And I remember I was talking to an apple farmer and he was going to give me some apples to graft onto my tree at home. And he was like, I'm going to give you some Macintosh. And I was like, oh, Macintosh, those are gross apples. They're mealy. They're just garbage,
Hmm.
right? And his comment, I found this really interesting, is he said that if you get the original type of Macintosh, it's actually nice and crispy and a really delicious apple. And so
Hmm.
basically what he was saying is that over time, Macintosh has been around for many, many decades, right? And basically over time, the quality of the Macintosh apple has declined. So we see this in other crops. And so to see it in cannabis it shouldn't be a big surprise. And
Right.
we found that, yes, there are mutations. Whether that's what is explaining the decline that people are seeing, I don't know, right? That's not really what we're saying, but it could be, right? The second one that you're talking about is epigenetics. And I think that's definitely a potential problem. And I would say that, the stress is one thing, but I personally lean more towards kind of aging and juvenility, right? And what we know about plants is that over time, plants start in a juvenile state. So if you look at, again, if we reference apples or any tree fruit, for the first 3 years, they don't produce flowers, right? Because they're in a juvenile state of growth, right? In cannabis, the juvenile state is only a couple weeks, and then they'll start flowering if you give them the right signal. But plants do have this juvenile state, and over time they reach a maturation. In cannabis, this is when you start seeing pre-flowers. It's a sign of they've gone through puberty. But what's interesting about plants in my mind is two things: is that one is that the juvenility of a plant is not uniform within an organism, right? And so if you look at a tree, the part of the tree that's closer to the trunk expresses more juvenile traits than the part further out, right? And in some plants, you can see this in leaf morphology and things of that nature. So that's one point. And the other point is that it's partially reversible in plants too. So if you have an older plant, there are things you can do to actually rejuvenate it and go back to a more juvenile state. And one of those methods is tissue culture. So that's one of the big, people talk about this reinvigorating your plants through tissue culture, right? And in the case of cannabis, we've seen some pretty good evidence of this. So a cannabis seedling starts with opposite leaves, and then eventually it goes to alternate leaf arrangement. What we see often when we come out of tissue culture, often they revert back to opposite leaf arrangement. They stop producing pre-flowers, and they revert to a more juvenile state. We even did some experiments where we were taking measurements of the leaf morphology, and in cannabis, the young leaves tend to be fatter and shorter. The leaf width to length ratio is different when they're older. So as they age, the leaves tend to get longer and skinnier. Juvenile plants have shorter, fatter leaves, right? And what we found is when we produced plants through tissue culture and compared them to clones of the same cultivar, they had shorter, fatter leaves again, right? So they had an opposite leaf arrangement, shorter, fatter leaves, and more juvenile traits, right? And so I think as far as epigenetics go, I think it's probably more related to juvenility,
That
like the decline. So as the plant ages, it's getting old and decrepit, right? And it's losing its vigor, right? The other thing we know with plants is when they're in a more juvenile state, they grow more vigorously, they root better, and I think that's going to impact growth, yield, cannabinoid content, everything.
So when you're taking clones are going to be the same age as their parent, right? So if you cut a mother, the age of the genome of the mother is as old as it is, and the resulting clones are going to have they're going to be the same age, right?
Chronologically, yes, but developmentally, no. And
Okay,
that's what I'm saying. So again, if you look at a tree, even within a single tree at any given time, the developmental age of different parts of the tree are not uniform.
so depending on where you take your clones from too.
Yes. Yeah. So you usually like when we look at perennials, the closer to the trunk you go, the more juvenile the tissue. It's
And so
counterintuitive too. Like most people would think that part on the outside of the tree would be the most juvenile because it's the youngest chronologically. But what we see is that developmentally, the tissue closer to the trunk is actually more juvenile.
Why? Is it because that's just how it is? Or is it because it doesn't get as much sunlight? Or is the plant allocating more resources towards the edges rather than the closer in? Do
But I'd
you
say, I'd say ultimately it's epigenetics, right? And so we have slightly different genes expressed on the interior. If I were just, this is speculation, obviously. But if you're a tree, the your mature tissue is where you're going to produce fruit, right? And you probably want to produce your fruit where you have more sunlight, more
Mm,
resources. Animals will see it and pick it up and distribute your seeds, all that kind of stuff. So evolutionarily, that's what I would expect. But I don't
mhm.
know 100%. I'm making that up. The other thing is that if you coppice a tree, if you just chop it down and it sends out a bunch of new shoots, you
Yeah.
want those to be young and juvenile to replace the mother plant, right?
Okay.
And so, I think if you wanted to, like from a cannabis, going back to cannabis, I think if you wanted to maintain juvenility, you shouldn't be taking cuttings from the very tip. I know that's what people do, but if you want to maintain juvenility, I don't think that's the best practice. It'd probably be better to chop the plant down, let it send out new shoots, and take those as cuttings. I'm not saying that's the best commercial practice. I'm just saying from a biological perspective. Or if you wanted to reinvigorate your plants without going through tissue culture, there's other ways you can do it.
Okay. And so when you would so you would recommend that if you're doing you just said this. So you're recommending that if you're doing clones from a mother plant, you would take from the inner branches and not from the outer branches. And then have you seen a difference in mutations from inner branches versus exterior sort of end of branch?
Is we have not got that far. I mean, that'd be
Is
great to
it
do, but we haven't done it. I guess what I'm saying is like in a standard setting, if you're not having any issues, I would take the cuttings from the top like people normally do. I
Mhm.
would say if you have a plant that's showing a pattern of declining, my suggestion, and I'd love to hear if this works, but my suggestion to you would be to cut the plant down and let it send out new shoots from close to the trunk and use those for taking cuttings.
Okay, so
'Cause-
that brings to mind like monster cropping, which is basically like cutting the plant down, which I have accidentally done. Like I've killed a plant on purpose. I cut the whole thing down and then lo and behold, it's not today. And it'll start growing again. So would that sort of practice that people do decrease mutations and I don't know. I don't know how to explain what my brain's thinking right now.
Yeah, like as far as mutations goes, like they did some studies in trees and they found the higher up the tree you go, the more mutations you find.
Okay.
We only took a few samples when we did the study in cannabis because it was very expensive and our budget didn't allow us to do more.
Mhm.
We didn't really see a clear pattern that way, but the reality with this plant is we didn't actually know the pruning history of the plant, and we can't really draw any major conclusions that way. But when they look at other trees, what they find is essentially that the further up the tree you go, the more mutations accumulate. And that makes sense biologically because they've gone through more cell divisions, right?
Right, because
And
that's
so,
the youngest, and so it's been generated more times.
what we would expect is you should have fewer mutations close to the trunk and it should be more juvenile. So it actually would be better in both ways. But again, that doesn't mean that it's commercially, I'm not saying that everyone should start propagating this way commercially. I'm just
Right.
saying that if you're having problems with a plant declining, I would start looking at some of these techniques.
Okay, to try and right the correct the what's going on.
Yeah.
Okay, that's first of all, tissue culture is super interesting to me. I'm just starting to learn about it, and epigenetics, as I said, has always been really interesting to me. So, the other thing that you are or have been involved with and have been doing for a while is polyploids, which also are super interesting to me. So, let's talk about polyploids.
Yeah. I'd love to.
So, I know you've created some triploids and tetraploids, and you've been looking at sex determination mechanisms in polyploids where they have XXY or whatever. So, let's talk polyploids because that's another super interesting topic.
Yeah. No, it's one of my favorite topics.
Good.
So I think there's a lot of unknowns with polyploids in cannabis, but
Mm.
I guess just for anyone that's listening that's not familiar, basically polyploids are any case where you have more than two copies of each gene. If you have a triploid, it means you have three copies. If you have a tetraploid, it means you have four copies. If you have more than that, then it just keeps going. So your hexaploid has six, octaploid has eight. A lot of the plants that we eat, so a lot of plants in agriculture are polyploids. So, some examples are strawberries are octaploids. So they have eight copies of each gene. Wheat is a hexaploid, and the list goes on. And so,
Potatoes, peanuts, cotton.
yeah, it's funny because a lot of the plants we cultivate are polyploids. In fact, it's disproportionately common in horticultural crops. And basically, what this suggests to me is that polyploidy has impacts on plant growth and development that are good for agriculture, essentially, right?
And are they polyploids that we created, or are they natural polyploids that then became very attractive to us because they have these extra chromosomes and therefore extra sugar, extra starch, extra big, you know, which came first, the chicken or the egg? Was it? Because I think
Well,
we developed the strawberries, right? We did that to the strawberries, the octoploidy.
Yeah, I mean, it's definitely a mix though, right? So some of them are natural polyploids, some of them are polyploids we created. I would say the majority of them we probably selected for without knowing it. Before people were intentionally creating polyploids, like polyploidy happens naturally. And so, basically we were going out in the fields and saying, "Oh, look at that plant with really big flowers. That's a really
Mm-hmm.
cool plant. I'm going to multiply it," right?
Mm-hmm.
And so, unknowingly we're selecting for polyploids.
Mm-hmm.
How I suspect it happened, right? Into the modern era, we've been doing it on purpose. So, a good example is watermelon. Seedless watermelon is a triploid, and
Mm-hmm.
that was created intentionally. Seedless banana was developed unintentionally many, many years ago. So people found this banana without seeds, and they were like, "Oh, that's really cool. Like, that's way better than a seedy banana." And so they started multiplying that, right? But then once we understood the mechanisms and how we could do this, we were like, "Oh, we should do that with watermelon too." And when I say we, I don't mean we. I just mean someone came up with that idea and implemented it.
And the cool thing about triploids is that they don't have seeds. They can't. They're sterile, right?
Well, that's a theory,
Pretty.
but
I mean, you still get, even in seedless watermelon. Sometimes you get little white little seeds that
yeah,
don't look. If you tried to plant one, and it wouldn't grow.
yeah. So the problem though is in cannabis, it doesn't follow the rules all the time. My
Cannabis.
joke is that cannabis has not read the textbook. It doesn't know that it's not supposed to have seeds because what we see in triploids, and this is one of those things that one of the problems is you have people that are trying to push these commercially, and
Hmm.
maybe they're exaggerating how seedless they are. What we see with triploids and cannabis is that some of them are pretty much seedless, but some of them are very seedy. And so
If
we've
you
had
let
some
them.
triploids that I would say are just as seedy as a diploid. But then we've had others that are pretty close, just like a seedless watermelon, where they only have a few husks, even when they're
Okay.
heavily pollinated. So, it's there's a huge variation. Why that is the case, nobody knows. But for some reason, cannabis is able to overcome this. So, the theory is that if you have three copies of each gene, you can't divide three in half when you're going through meiosis. So you're trying to make pollen and eggs, and you can't divide three in half. The theory is
But
that that should make them sterile. But what we see in cannabis is that it can overcome that somehow. And we've tested the ploidy of some of the progeny. So when you grow out the seeds from a triploid, what we found is they're anaploid or disploid. We're not sure if it's anaploid or disploid. That's just a technical difference. But basically, they're neither diploid or triploid. They're just somewhere in the middle. So they have extra chromosomes. Maybe they're missing chromosomes. They might have extra pieces of chromosomes or missing pieces of chromosomes. We haven't gotten that deep. But what we do know is that their DNA content is somewhere in between where they should be, right? And so we don't know why, we don't know how, but that's the case. But then other triploids are pretty seedless. So I think long term, we'll definitely be able to develop seedless triploid cannabis. But right now, it's kind of hit or miss. And I think
so
there's a
it's sort of,
couple,
there's no
there's
guarantee that your triploid that you're purchasing from whatever company is guaranteed to be seedless. It's you're kind of taking a roll of the dice there, because
yeah,
they
and
might.
there's a couple field trials from some American groups. So it's triploid hemp, but same species. And basically what they found, I think on average, if you look across the different field trials, so there was one from one of the authors was from Oregon CBD where they found that it was pretty much seedless. But
Mhm.
then there's been a couple subsequent field trials where they were testing triploid hemp, and on average they were producing about 30% as many seeds as a diploid. So I often say it's kind of like stainless steel, right? It stains less, but it's not stainless, right? It still rusts. And so it's seedless, not seedless.
Okay, that makes sense.
But I do think long term we'll be able to, once we understand a little bit more about this, do a little bit more breeding and selection and stuff, we'll be able to develop seedless triploids. But until then, you have to be careful, is all.
And so it's not like you can't just make a seedless variety, a triploid seedless variety from any strain. Like there are going to be plants that are better at being seedless,
Yeah.
and there are going to be some that are better at keeping on reproducing like they want to do so badly.
Yeah. And we've grown some of the seeds from triploids, and they get pretty wacky. Like I said, they're aneuploid, and we see a lot of like irregular leaves and a lot of mutations and just a lot of odd growth habits.
Mhm.
So, and
Okay.
not in a good way, right? Like, so the aneuploids don't tend to be, it's not a good weird. It's kind of like they tend to be runty and just, maybe there's some goodness in there somewhere, but it's generally
All
not good.
all gnarly and weird.
Yeah, and a lot of like we had some that like instead of having serrated leaves, they were just smooth leaves. We
Mhm.
had some that were, we had some with odd numbers of cotyledons, a lot of fasciation, things like that.
Interesting. So fasciation could be associated with polyploidy, but
Well, those
is
are
it?
aneuploids, so
Oh, okay. All right. So, yeah, okay. Polyploidy is, and then what about the tetraploid? So we talked about triploids. What is the benefit of a tetraploid? And I would imagine that those can produce seeds because the four and the two can happen. So, yeah, give us the DL on the tetraploid.
yeah. So I mean, the main use of a tetraploid is to produce a triploid. So the way you produce a
Mhm.
triploid is by crossing a tetraploid with a diploid, right? So most people are using tetraploids specifically for the creation of triploids. But that being said, I do think there's potential of tetraploids. In general, they tend to have bigger, wider leaves, but when they go into flower, I find they're pretty similar, maybe a little bit different. But I think the one thing we have to be careful with, and this is again one of the things that I think everyone needs to be aware of, is that the impact of polyploidy on one genotype might not be the same as on another genotype, right? And so when you double the genes, it's going to accentuate the genetics, the genes that are present in that individual. And so if you polyploid two different plants, you could see very different outcomes. But where I see the real benefits of tetraploidy is kind of when you start crossing tetraploids. So again, a lot of the papers, and the devil's in the detail a lot of the time. But a lot of the papers that look at tetraploids, they take a diploid, they turn it into a tetraploid, and then like a clonal tetraploid, and then they compare the two. And the problem with that is then you have the same genotype, except that you have twice as many gene copies,
Mhm.
right? And so you're looking at the gene dosage effect, but you're not really doing anything else. I think where the real beauty of tetraploids are going to come in is when you start crossing at the tetraploid level, because
Mhm.
then you have the potential to have, instead of two different copies of genes, you have four. So you could have four different versions of a single gene and start getting some really interesting phenotypes, right? But
Mhm.
if we look to other species, like sometimes tetraploidy can affect like water use efficiency. So you have fewer larger stomates. So maybe you can survive droughts better. Sometimes they're more disease resistant. Who knows, right?
Is
But again,
there
I
a
think
difference?
it will be very genotype specific.
Is there a difference, or do you know if there would is has been observed a difference in like phytochemical production or yields or things that, all these cultivators are scrambling to, have?
Yeah, I'd say nothing consistent. So in general, like sometimes you'll see an increase in cannabinoid content. Sometimes you won't. Sometimes you'll see a difference in terpene content. We measured some trichomes. So we saw larger trichome heads, but I wouldn't say there's anything like it's not that consistent, but
Not earth shattering?
no. And I think one of the reasons for that, like they see this in a lot of species, but I think in the case of cannabis, we pushed the plant pretty far, right? Like,
Mhm.
how much as a percentage can a plant really produce?
Yeah, and what's your thought process on that?
Well, I first of all, I think a lot of the numbers are very generous that are out there, and I think the data supports that, right? Like when people take commercial products and test them, I'm sure you're very well aware of this. I believe some of it was probably from your research
Yeah.
actually, but the actual values in the jar is much lower than what they're putting on the label. I think there's a number of reasons contributing to that, but so I don't really believe that all plants are like 30%. I think it's pretty rare to get a plant that's over 30% legitimately. I'm not saying it's impossible, but I don't think that's as common as they would have you believe. I think getting much above 30 is a bit of a stretch.
Okay, so your threshold of skepticism lies around 30%.
Yeah, I mean, I think if you're breaking the 30 barrier, then that's pretty exceptional. Much above
Oh,
that, like,
yeah. My rule, I don't know where I heard this from. Some people have said Daddy Mary said it, but he says that he didn't say it, but 35% is like the max. It is, so basically over 30% is exceptional. Over 35% is basically unobtainable or biologically impossible. Um, that being said, I do have to think about the structure of cannabis and where those cannabinoids are made and stored in the trichomes, right? So if you have a larger trichome head or you have more of those trichome heads, could we potentially push it over that just based on trichome number or trichome size?
so where, what I usually come back to on this whole discussion is that, like, if you look at hash on the market, what percent is it? Maybe 60, 70%, right? So how can, like, isolated trichome heads be 60 or 70%? If that's the case, how could you ever have a plant that produces more than like 35? I just, it
Yeah.
just logically, it's hard for me to wrap my head around, right? Because, like, when you have hash, which is just trichome heads, maybe a little bit of leaf plant material in there, that's 60 or 70%. So, how much of the plant is actually trichome head? I don't know.
Yeah. I don't know either, but these are the things that keep me up at night.
Yeah, I also, yeah,
Not that I want to push any plants over. I think 35 is that's kind of the biological threshold for the plant. And I honestly, like, if you want to get higher THC, then probably flower is not what you're looking for, right? I feel like there's always trade-offs in biology. If you're pushing something to go this way, then other things are going to suffer because of it. Like if you're working really hard and you're not getting any sleep and you're stressed out, your health is going to suffer. Right. And I think that it's the same, this is the living world. If a plant is being pushed to produce higher and higher phytochemicals, they're not putting energy towards fighting pathogens and things like that, and maybe they're not gonna be as healthy, and I think I've observed that plants with higher THC are more disease susceptible, um, and I don't know if there's been any extensive research on it, but that would be something interesting to do. You got any room
yeah,
for a postdoc?
yeah. We have room. It's more about funding than space.
Yeah. Well, I'm available. Okay, so I heard you say that most mutations are bad. When you were talking about Mueller's ratchet, most mutations are bad. I was always taught in school that most mutations are neutral. They're neither bad nor good. They just kind of slide through,
Okay.
right?
Yes.
So are you saying that if we're just looking at good and bad mutations, most of them are going to be bad? If we're looking
Yeah,
at
I,
good
I would
mutations
agree with what you're
overall.
saying. I would
Okay.
say most are probably neutral, but then if they have an impact on gene function, it's probably going to be negative.
Okay. All right, cool. Because I was like, "Oh shit, everything I know is wrong." So, let's see, is there anything else on my list here? Oh, the polyploids, the sex determining mechanisms, right? So if you've got a triploid, okay, so cannabis, most of it is either female or male. We've got X chromosomes, Y chromosomes, females are XX, males are XY. Sometimes there are exceptions, but when you've got a triploid, you could have XXY, XYY, XXX, not YYY.
No.
That wouldn't be viable. So, when you were looking at sex determination mechanisms in polyploids, what exactly did you look at and what did you find?
Yeah. So when we did that study, crossed a female tetraploid with a male diploid, right? And so we ended up with a population that were XXX or XXY. So we didn't have any XYYs in the mix. That would have been hard to generate, I suppose. And basically, what we expected to find, or what I expected to find, is that what we were hoping to find, I guess I should say, is that XXY plants would be female but have a little bit of, the males are always a little bit vigorous when you start growing them. You can usually pick out the males because they're the biggest,
Hmm.
largest plants. So what we were hoping is that the XXY plants would be anatomically female, but have a little bit of boosted vigor from the Y chromosome, right? That's not what we found. What we did find is that the XXY plants were kind of a spread. Some were pretty much female, some were pretty much male, and then there was a whole diversity in between, right? And so what surprised us about that is that I would have expected that XXY plants, like if they're going to be hermaphroditic, I would expect them to be kind of uniformly hermaphrodite, right? And that's just not what we found at all. What we found is that the XXY were just this huge gradient of plants with really diverse sexual expressions. We also found some of the individual florets were kind of weird. So sometimes the flower itself would be hermaphroditic, which is unusual for cannabis, right?
Hmm.
And sometimes they'd have odd numbers of stamen or stigmas, things of that nature. But what's really interesting about it is that the XXY plants had this gradient, which suggests that there's more at play than just the X and the Y chromosome. And
We already know that.
realistically, yeah,
We are,
we already knew that because you can turn a female into a male even though there's no Y chromosome,
so there's
or vice
hormones
versa.
at play and receptors for those
Yeah,
hormones.
yeah, and so that's basically what it came down to. And but it was quite interesting. But that's just not what we expected. We expected the XY ratio to have a similar effect.
That is kind of interesting that you did get that whole spectrum of sexual expression.
Yeah, we
Cool.
also, we also had some of the plants would have, one branch would be male, one branch would be female. So it
Hmm.
was kind of interesting. And we suspect this is related to the growth regulator gradients within the plant and stuff like that.
So you
But
just
we don't,
got the whole gamut of sex.
yeah.
Very cool. How many plants did you grow out from that?
It was not a large population. Our license at the time didn't really let us grow that many. So I think we only had like 20 something plants,
20 plants, 20 something. Okay.
somewhere in there. So yeah, it was a small population. And, but even within that small population, we saw this huge spread. So
Right. It's not like if you had more plants, you would see less diversity. It would just sort of maybe be even more blended.
yeah, I mean, in an ideal world, we would have had a large population and all that kind of stuff. I know when I talk to other like breeders of other crops, they're used to talking about plants in the tens of thousands when they're doing breeding projects and stuff. And when you have to justify why you're only working with a couple hundred or like a couple dozen or something,
Right.
the classical breeders are like, well, that's not a big enough population. And I know that that's very true.
Hmm.
But that's the constraints we live with.
And and are those constraints still just as like hard to like? Has it gotten any better since it's been
Yeah.
so many years now in Canada with it being legal and you guys can research it and stuff like that? Is it getting better?
Yeah, it's definitely gotten better. And so when it was first legalized, the security requirements were quite serious. And if you wanted to grow a large number of plants, you have to have like motion detectors and cameras and all that kind of stuff. Since then, they've dramatically reduced the security requirements, and
Hmm.
so we have a lot more freedom to operate now. We have an institutional license, which allows us to grant sub licenses within the university. So what that means is that if I want to, let's say I have a greenhouse space that I want to grow some cannabis in, I can do that internally at the university instead of having to go and request a new license from Health Canada, which just means that we can do it quicker. We can do it without any, we have to follow the security matrix and stuff that we set out,
Hmm.
but it's much better that way. And so we've recently redefined our security matrix, which will make it much more practical. So if we want to do greenhouse studies, for example, it's actually possible now, whereas in the past, there was no way we could do a greenhouse. It just wouldn't have worked.
And so do you have greenhouse space and is there like a plant count that you're allowed like a max, or do you just kind of do whatever?
So, I mean, we have greenhouses here, but none of them are licensed right now. So if I wanted to do greenhouse, I would have to go through the process of getting an internal license, which could be done fairly quickly and reasonably. And then it's not based on plant count; it's based on square footage.
Okay. And would the greenhouses be shared? Because I think I've read some studies, and I'm not sure if they were out of Canada or I think it was out of Canada, where they would have only, a small number of plants that they were growing out to, look at whatever they were looking at. But the issue was not that they couldn't do more. The issue was that the tomato guys took up the whole greenhouse and only gave them a small area to work in.
Yeah, so I mean, I'm lucky where I am that we have a lot of greenhouse space, and essentially the way it works is we just have to rent it internally. And so if I want space, then I have to rent it, and we would never be able to share space with a tomato guy like that. That's
No.
not how it would work. No. We have to control access and all that kind of stuff, right?
Yeah, yeah.
But we're very fortunate that space is not really the issue. We if we wanted greenhouse space, we could do it. We would just have to license it. So there are security requirements. We'd have to get that approved, and then we could do it.
And do you have to grow indoors, or could you do outdoor field trials as well?
Theoretically, we could do outdoor. It's just challenging because there are still some requirements, and the most practical on that side here would be there's some rooftop space, so we could do it in pots outdoor. But if we want to do in the ground, then we would have to find space. For that kind of research, we would probably have an easier time just collaborating with a licensed producer, and they're
Because
most of them are
you'd have
quite
to have
collaborative.
security. You'd have to have security and stuff. I would imagine if you were growing THC type cannabis outdoors,
Yeah,
where students can just come along and pick their dinner.
yeah. We have like a security matrix, and we would have to follow that.
Yeah.
But more importantly, like on main campus, we don't really have any outdoor space dedicated for
Yeah.
that type of stuff. There's maybe some places we could do it, but we do have agricultural research stations. But then it would get complicated.
Yeah. And collaborations are always nice. Other than the whole thing that we constantly get accused of is, biased science. We're getting paid by big ag or big cannabis to fudge the results and make up the statistics and get paid to produce the results. Maybe you should talk a little bit about that because I'm sure you get it too. People think that we take all this money and are rich and because we're getting paid by, whoever to generate whatever results they want.
I don't know that anyone's ever actually said that to me, but I'm sure people think that, but I've never really come across it very much. Most of the funding in Canada is tied to collaborative research with companies. So most of my research is with a company.
Hmm.
So I'm sure people probably have that thought, but that's just how it's structured.
Yeah. Yeah. I can. And the thing is like, I'm sure there are scientists who are paid by there are scientists who work for Cargill or any one of the large agricultural. They hire a suite of scientists to do field trials, to do all of this genetic work to figure out problems, but they don't publish those studies. That's in-house R&D. Whereas you are doing research through an academic institution sh- should be relatively unbiased because you're not working for this company. You're working with the support of this company where they want the answers too.
Yeah, and I think it probably depends on the type of research you're doing too. Most of the research I do, I don't know that that's really like, it's not like we're making claims about a product usually.
Hmm.
So I don't know, but
Is do you do proprietary work with some of these companies? Like they want to do R&D on polyploids or tissue culture or something, and then you do the research, and then they take the outcome and kind of run with that, and then you don't publish it.
yeah, we can. Usually that would be like contract research. So,
Okay.
we like if a company was interested, they could come to me and offer me money to do research at the university, and we would do it. The university charges overhead,
Yes.
and we don't have to publish that kind of stuff. And yeah, we do some of that, but in that case for us, it's usually the interest there would be we're backing up some of this plants in tissue culture, and we do some of that. So if someone wants to collaborate in that way, we can do that. But more often, companies are willing to apply for grants where we leverage their funds and then do a research project.
Okay.
That's usually what we do. But
And tissue culture, like it's a big conversation right now, especially in cannabis. Everybody wants to do it. Everybody wants to have it. Everybody's talking about it. How easy is tissue culture, and how profitable? Like, if I wanted to open a tissue culture lab tomorrow, is it a good idea? Is it a bad idea? Is it an okay idea? Like, is yeah, like what are your thoughts on it as a business? Because I feel like every day I'm find another tissue culture lab that's opening, and for various reasons, whether it be a library or a bank or pathogen cleaning services, and or all of the above.
yeah, I mean, I don't really, I'm not much of a business person,
Me
so I
neither.
don't know. I don't know if it makes business sense, but I'd say like, when I first started working, there were a lot of companies investing a lot of money in tissue culture. And at the time, the methods really weren't that refined. And
Mhm.
so the methods that were out there were not very reproducible, and a lot of people lost a lot of money doing that. They were investing tens or hundreds of thousands of dollars setting up a tissue culture lab. Then the methods didn't work, and then they shut it down. I think the other thing about it is that it's really important to understand like what tissue culture can and can't do. And so tissue culture, people like me are our worst enemy because, we'll go and make claims that, oh, you can produce millions of plants using tissue culture. And that's technically true, but, it takes a long time to ramp up to that. And cannabis doesn't really like tissue culture, right? So cannabis is this vigorous plant that grows like it's a beast in the growth room, but it's a delicate princess in tissue culture. It's really finicky.
Yeah.
And, now we're to the point now where, we can quite reasonably, like we probably have like 200 or 300 different strains in our culture room at any given time. And, we have trouble with a couple of them, but most of them respond pretty well. But that being said, like it still, to me, doesn't make sense to mass produce plants through tissue culture. Makes more sense just to maintain clean plants in tissue culture and replace your mothers once in a while that way. If that's what you want to do, then I think it's quite reasonable to set up a tissue culture lab in-house. And I think it's quite reasonable that you can set up a company to do that. And, you can do tissue culture with some fairly basic equipment. It doesn't have to be super fancy. It doesn't have to be that expensive. There's a couple key components that really matter. But if you know what you're doing, then you can do it for a reasonable cost. And I think it would be practical.
And you did write a chapter in a book. We were both authors in this book called The Cannabis Genome. And your specific chapter was specifically on tissue culture,
Right.
setting up a tissue culture lab. Is that right?
Yeah, that's correct.
So what all did you put in there? Because I haven't read it yet.
Well, I mean, most of it's fairly standard tissue culture lab setup in general, and so it has the different components. And there's some key components to setting up a tissue culture lab. So you have to have your transfer room, you have to have your culture room, you have to have your media prep room, and how you lay those out matter. And so typically you want to essentially set up your lab so that you have positive pressure, so that the air is moving from the cleanest part of the lab to the dirtiest part of the lab, right? If you were to visit my lab at the university, that is not how it's set up. And so, that chapter is really about like, if you're building a lab
ideal.
from nothing and you have the option to do all these things, this is what you should do. But I guess my point with this is that, that's best practices, but you can get away with other things, right? And so in a university world, when I first started here, they're like, okay, this is your lab space, and it's not tissue culture. It wasn't built for me specifically for what we're doing. And so I basically had to retrofit space to make it work, right? That in that chapter is like, that's what I would love to build, but it's not what I have.
And is that so because you don't have the ideal situation of having the flow go from the cleanest to the dirtiest part of the lab, do you have problems with contamination issues, or does the hood take care of most of that?
So we have some contamination. Everyone has some contamination. Whether it's a problem, it depends. We'll go through periods where we will have problems, but in general, I'd say not really. We rely heavily on, we have a lot of HEPA filters in our space. So we filter our air quite heavily. We leave our flow bench running to filter the air. We try to be, we change our shoes and everything going in and try to be as clean as possible. I'd say the biggest issue we've had with contamination is actually related to thrips. And so thrips are little devils. Thrips are fairly reasonably, you can manage thrips in a growth room or a greenhouse. But the tricky part from a tissue culture perspective is that you can bring plants into tissue culture, and sometimes thrips lay their eggs inside the plant tissue. So when you surface sterilize the plant, you can actually get thrips that survive the process. And what they do is they actually, they'll hatch and they'll start growing inside your tissue cultures. And where it gets really tricky is that they can actually crawl out of a box and into another box, right? And so it's not actually the thrips that are the problem, but when they're traveling from one box to the other, they'll pick up spores or whatever, and they spread contamination. And so the worst problem we had with contamination was because we had a thrip outbreak and they started spreading microbes. So we had fungal contamination spreading from box to box explosively. And so that was a big problem. And HEPA filters aren't going to do anything about that. Washing your hands isn't going to do anything about that. Nothing will do anything about that. The only thing that will do anything about that, and so we've implemented since then, is a very strict quarantine process. So when we bring anything into tissue culture, we quarantine it, and we've implemented putting it on an orthene-based media. And orthene is a systemic insecticide that will kill thrips. And so we quarantine it on orthene, and then we bring it into the general population. The only time we don't do that is sometimes we're working with licensed producers, and they don't want the risk of orthene into their system, and so we'll bypass that and we'll keep it in quarantine for a longer period.
They get their own house that yeah. So that's
Well, orthene's not an approved pesticide in cannabis production, right?
yeah, no, I get it. So in that case, like let's say I'm a super granola hippie and I don't want orthene, so you bypass that particular method. And so then I would have a designated area for my hippie plants. So if there is an outbreak, it can't get into any of the other ones.
Yeah, exactly. So we basically, because of regulations, we can't physically isolate them. So we don't have the space to have a quarantine zone. So we
Mhm.
bag them up. So when we first bring them in, we bag them up and we have little sticky traps and we monitor that very closely. And once we're convinced that they're clean, then we'll release them into the Gen Pop.
I gotcha. Okay, cool.
But
That's super interesting. Because the call is coming from inside the house, right? It just Trojan horse situation. They're in there,
yeah,
and then all of a sudden, they're all in everywhere.
yeah.
That's
And if you talk to people that have done tissue culture for like, later in their career, most
mhm.
of them have had like one or two events of catastrophic loss because of insect getting into their culture room.
So even okay, so that's I mean that's good practice to have with when you're taking a nursery intaking clones. Like you should have a quarantine area to make sure that the clones that you're bringing in don't have something like thrips or hot plate viroid before you put them into your general population. So that's it's a similar sort of situation with the tissue culture, which is
It
interesting.
is. It's. I guess the main difference is most people like the assumption with tissue culture is that the surface sterilization would take care of insects, right?
Hmm.
So most people don't really think about it, myself included, right? I learned the
Right.
hard way,
Yeah.
so I like to share this so that other people can learn from my mistakes instead of their own.
Well, hey, thank
So
you.
if you're setting up a tissue culture lab, yeah, watch out for thrips.
Okay. Wow. Wow, I wouldn't have thought of that either until something really bad happened. So what else should we talk about? What are you working on right now?
So like I said, we're doing the meristem culture. That's the latest project that we have it up and running. I have another student working on comparing extractability of different strains, and so the variability in, some plants are good washers, some plants are okay for rosin, some are better for dried flower. So we're looking at the variability in extraction efficiency, and that's one
Mhm.
thing we're doing some cryopreservation work. So we've done some cryopreservation in the past, and what we found is it was very genotype specific. So we're trying to determine why that's the case,
Mm.
trying to refine that.
So I've heard so I'm going to interrupt you cuz I just something came to mind. I have heard that tissue culture, back to the tissue culture thing. Tissue culture is also strain specific. Like it could be that some strains require a different hormone mixture than others in order for them to survive and get brought out of or what did you call that? Climatization?
Yeah.
Go through the climatization process. Is that correct?
Yeah, so I mean that's true of any plant species where you'll see variability between genotypes.
Mhm.
Um, we largely don't use growth regulators for our tissue culture, which is kind of
Mhm.
unusual. And like I said earlier, cannabis has not read the textbook. It doesn't know that it's supposed to need growth regulators. But what we found, and we tested like 5 different cytokinins at like 4 different concentrations each, and none of them were better than the control. And so, if you read through a textbook, a tissue culture textbook, it will say cytokinins increase branching and bud break and multiple shoot formation, etc. And what we found with cannabis is that's not the case. And we more or less don't use growth regulators. And I think that's really important for this, like when you're talking about genotype specificity, is that in our case, like I said, we probably have like 200 or 300 different genotypes, and some of them perform better than others.
Mhm.
But I'd say that we don't actually change the method for every individual cultivar by any
But
means.
they're all in the same media and just some do better than others and there's no real reason to have multiple types of media for different strains because they all pretty much do well in this control, no hormone, no plant
Well,
growth.
my feeling on it actually is that genotypes are probably more variable in their response to growth regulators than they are to tissue culture itself. So when you start adding growth regulators, some won't like it at all, some might tolerate it. And so when you start adding growth regulators, I think you're going to run into more problems. So you're kind of creating your own problems in a way. I found for cannabis, They do better in tissue culture the less like tissue culture you make it. And what I mean by that is that the rule of thumb in tissue culture, the standard light level is like between 40 and 80 micromoles of light.
Hmm.
I found they do much better if you crank the light up, sometimes up to like 300 micromoles, which is for tissue culture, that's really, really high. For normal plant growth, that's not very high, but for tissue culture, it's really high. They do really good if you increase ventilation, increase light, and basically, like I said, make it less and less like tissue culture. In general, I find cannabis in tissue culture, if it forms roots, it grows a lot better than if it doesn't have roots. And so I actually think if I were interested in larger scale multiplication, I would probably look at more of a hedging technique. So you have a plant with roots and let it grow, decapitate it, use that for multiplication, then let it send out new shoots. And that's kind of the opposite approach that tissue culture usually uses. But for cannabis, I think that's a logical approach.
Yeah. All right. Interesting. So let's see. What else do we want to talk about? You got any other passions, interesting conversations online that you and I are often involved in the same one?
Yeah, I don't know. Not sure. Like we've covered quite a bit of ground.
Yeah. All right. So you've got no more words of wisdom on cannabis, cultivation, research. And I guess we could probably just wrap it up. It was so good to talk to you. Like I said, I'm still learning about tissue culture, and it's nice to have somebody who has the expertise in it, such as yourself, to ask questions to and whatnot. And you are definitely doing research that I'm super interested in, all of the things.
Well,
So,
thanks for having me. It's been a
do you have any
pleasure.
questions? Do you have any questions for me or um
Well, so what are you up to now?
me? Oh, well, I'm working with Angel Fernandez at MyFlora DNA. That's part of the reason why I need to learn about tissue culture and some other things that I'm not versed in. Teaching, going into our, I think, 21st semester of teaching cannabis science at the University of Colorado with Daniela Vergara. I am, what else am I doing? We're starting a company called TrueCut, which is where it's a software company at the base of it, but we're doing genomic fingerprinting to bring sort of market maturity to the cannabis industry for people to register their strains, to license out cultivation, breeding rights, to for cultivators to verify what they're growing. So there's cultivators out there that are like, I don't know, I think this is Durban poison, but I mean that's what they told me it was, but it doesn't seem like it is. So can you tell me, is it or is it not? Create, try to help breeders find interesting things that they might be wanting to seek out to do different crosses, seeking out unique genes or genotypes for unique phenotypes. And then we want to connect that. So, all the way from identification, verification, and then bring that to the counter. So, when somebody is cultivating something that has been true cut verified, then the consumer can see it has been true cut verified. It is what it is on the label. And then Riley Kirk is on our team of folks, then we can start asking consumers what they experienced with a true cut verified. So I had Durban poison that's true cut, Durban poison true cut verified. What was your experience like? So then maybe we can start connecting strains with
Right.
experiences. And of course, we realize that something in California might be different than New York just because of the environmental conditions or whatever. But at least if you're starting with the same genotype, you're going to get much closer to the same phenotype than if you've got two completely different genotypes just called the same name. So we're hoping that we can, and then the consumers can say, okay, I've had this, it's true cut verified, it works for whatever I'm looking for it to do. And so I'm going to keep purchasing that as long as it's true cut verified. So that's kind of what we kind of want to close that whole circle and start bringing, some trust and some consistency to the market where there really isn't a whole lot in the United States at the moment. I don't know if it's any better in California or in Canada, but no
I
better
don't think so.
because there's still, you guys don't have a verification system either. It's just like it is what it is.
No, it's funny because our hemp is very much regulated and certified, but our adult use cannabis, it's the Wild West.
Okay, that's yeah. Well, the hemp types have been around for a while, right? So there's like lists of hemp that is verified because it's come from other countries or
Here, you can only grow hemp here if it's on the government approved list of hemp, and it has to be produced through the pedigree seed system. So it's very much
yeah,
regulated and validated and all that. But the drug type cannabis is just nothing.
whatever.
Yep.
Let them eat cake. Okay, yeah, so that's what I'm up to, and just, a couple of side gigs and just this podcast, webcast thing is gonna be taking up a lot of my time because the interviews take a while and then editing but I don't know, we're releasing an episode every Friday, so I'm not sure how far down the road this one's gonna be, but I've
Cool.
got some really awesome guests, yourself included. So,
That's great.
yeah, it
Yeah,
was really good
if
to
you're ever in
talk to you.
Ontario, let me know.
Yeah, I would love to come up to Canada and visit you and see what you got going on. Yeah. And
Yeah.
same, if you're ever in the United States, if you're going to any conferences down here, be totally awesome to hang out with you.
Well, yeah, if I'm ever there, I'll look up.
Yeah, great. That's awesome. Thank you so much for coming on. I appreciate your time and your words of wisdom and all that. And I'm sure that we'll keep crossing paths on good old LinkedIn.
Yeah, great.
All
Well,
right. Have
thanks.
a good rest of your day.
Yeah, you too.
All right. Bye.